A stopwatch clicks at a Kingston, Jamaica track meet packed to capacity with school children screaming for runners barely old enough to drive. 4,000 miles away, in Kenya's Rift Valley, a teenager runs 12 kilometers to school before sunrise, the same route his older brother ran to become a national champion. Two different scenes, two different specializations, explosive speed in one, relentless endurance in the other, and a question that has driven more sports science research than almost any other in the field. Is this really written in the genes of an entire race, or is something else going on that a simple genetic hierarchy completely misses? In a genetics lab, researchers pulled DNA samples from elite sprinters, Olympic weightlifters, and world record marathon runners, alongside ordinary people who've never set foot in a gym. The plan is to find the genetic fingerprint of raw physical strength and athletic dominance, population by population, and see just how big the gap really is between groups. What the data actually revealed wasn't the clean, simple story most people expect walking in. It was messier, stranger, and in some ways far more surprising than a straightforward genetic hierarchy. Stay with me, because the real explanation behind athletic dominance is not what most headlines make it sound like. Let's start with the gene most people have actually heard of in this conversation, because it's been the subject of magazine headlines for over a decade. ACTN3 controls production of a protein called alpha-actinin 3, or found specifically in fast-twitch muscle fibers, the type of muscle tissue responsible for quick, powerful, explosive movements like sprinting and heavy lifting. A specific variant of this gene, sometimes nicknamed in the press as the speed gene, comes in two versions. One version, carrying a functional R allele, produces normal amounts of this protein. The other, an X variant, results in a complete absence of alpha-actinin 3 in muscle tissue. People who carry two copies of the X variant, meaning they produce none of this protein at all, tend on average to show a slightly lower proportion of fast-twitch muscle fiber and a modest reduction in raw power output. Here's where the population data gets interesting. Researchers have measured the frequency of this non-functional X variant across a wide range of populations, and the numbers do genuinely differ. Populations with West African ancestry, including Jamaican, and many African American populations show this non-functional variant at strikingly low frequency, often in the range of just 2 to 4% of individuals carrying two copies. Kenyan and Nigerian populations sit even lower, often cited around 1%. Ethiopian populations sit somewhat higher, around 11%, closer to the range found in European populations. Populations with broader European ancestry sit meaningfully higher, somewhere around 18%. Populations with East Asian ancestry sit higher still, with some studies putting the frequency around 25%. On paper, that looks like exactly the kind of clean genetic gradient a headline like this one would want. But the actual research tells a very different, much more careful story once you dig past the headline number. And the first hint of that comes from the fact that Ethiopian and Kenyan populations, despite living in neighboring countries and both producing world-class distance runners, actually show quite different frequencies of this specific variant, which on its own should raise a flag about treating this as some clean, unified African genetic pattern at all. In 2010, a team of researchers directly tested elite Jamaican and United States sprinters, the two populations that have produced the fastest 100-meter runners in recorded history, and compared their ACTN3 genotypes against non-athletic control groups from the exact same populations. If this gene were really driving elite sprint performance the way the headline suggested, you'd expect elite sprinters to show a meaningfully different genotype distribution than ordinary people from the same background. The study found no statistically significant difference at all. Elite Jamaican sprinters and ordinary Jamaicans showed essentially the same ACTN3 genotype frequencies. The same held true comparing elite American sprinters against the general African American population. The researchers' own conclusion was direct. Because the non-functional variant is already so rare in these populations to begin with, there simply isn't enough genetic variation left for this particular gene to meaningfully separate elite athletes from everyone else in the same population. A separate study looking specifically at East and West African athletes found something similarly complicated. Testing Ethiopian endurance runners, Kenyan endurance runners, and Nigerian power athletes against non-athlete controls from the same populations, researchers found extremely low frequencies of the non-functional variant across the board in Kenyans and Nigerians, and no meaningful difference between elite athletes and ordinary people, even in the one population, Ethiopians, where the variant showed up at a somewhat higher baseline frequency. The paper's own stated conclusion was blunt. Alpha-actinin 3 deficiency does not appear to be a major influence on athletic performance in these populations at all. This is exactly why modern sports genetics research has largely moved away from hunting for any single athletic gene and toward what researchers call a total genotype score approach, combining dozens, sometimes over 100, different genetic variants, each contributing a small individual effect into one composite profile. Even with this more sophisticated approach, researchers in the field are careful to note that genetic testing on its own remains a limited tool for predicting athletic success, useful only when combined with actual physical testing, training history, and performance data. If a single simple gene, or even a modest handful of them, reliably explained elite athletic dominance, sports science would not have needed to develop increasingly complex, multi-gene, composite scoring systems just to capture a fraction of the picture. Here's the number that really deserves attention because it's the piece that gets left out of almost every popular article about this gene. Multiple independent research groups have estimated that variation in the ACTN3 gene accounts for somewhere around 2 to 3% of the total variance in muscle strength and sprint performance across the general population. That means, roughly, 97% of what actually determines an individual's raw power and sprint capability has nothing to do with this specific, heavily publicized gene at all. A single genetic variant that gets constant media attention as the sprint gene, turns out, under actual controlled research, to explain a genuinely small slice of the real picture, buried under years of headlines suggesting otherwise. There's another gene worth understanding here, precisely because it demonstrates just how individual rather than racial real genetic strength advantages actually turn out to be. Myostatin is a protein that normally limits muscle growth, acting as a biological brake on how much muscle mass the body builds and maintains. Rare mutations that disable this gene produce a dramatic effect. Individuals with a double copy of a loss of function, myostatin mutation, develop noticeably increased muscle mass and reduced body fat, a condition first documented in extraordinary detail in a small number of case studies, including a German child born with visibly enlarged muscles from infancy, whose family history traced the mutation through several relatives with unusually high strength. These myostatin mutations are exceptionally rare, have turned up in isolated individuals and families across multiple different populations and continents, and show absolutely no pattern of clustering by broad racial category. It's one of the clearest, most dramatic genetic strength advantages ever documented in humans, and it underscores exactly how these rare, powerful genetic variants distribute themselves at the level of scattered individual families, not along racial lines. Twin studies add another important layer to this picture because they let researchers estimate how much of athletic performance is even attributable to genetics of any kind, as opposed to training and environment. large-scale twin studies comparing identical twins, who share effectively all of their DNA, against fraternal twins, who share only about half, have estimated that somewhere around 60 to 70% of the variation in general athletic performance and physical fitness traits within a population is influenced by genetic factors. That's a meaningfully high number, and it confirms genetics does matter for athletic ability. But it's a heritability estimate that describes variation within a single studied population, not a number that tells you anything about differences between separate populations. A distinction population geneticists emphasize constantly because it's so frequently misunderstood or misused. Which heritability within a group says nothing about the source of any average difference observed between two different groups, especially when those groups differ enormously in training access, nutrition, cultural investment, and economic opportunity. So if one specific, well-studied, strength-related gene doesn't explain the gap in elite athletic performance, what does the bigger picture of muscle physiology actually show? Muscle fiber type composition, the ratio of fast-twitch fibers built for explosive power to slow-twitch fibers built for sustained endurance, does show real measurable variation between individuals, and that variation is influenced by a large number of different genes working together, not any single one. Studies measuring muscle biopsies in elite athletes have found some individuals with fast-twitch fiber percentages above 80%, and others, even among trained endurance athletes, with slow-twitch percentages just as extreme. And this kind of dramatic individual variation shows up inside every population. Researchers have studied closely enough to measure it properly. But here's the part researchers emphasize again and again in the scientific literature on this topic. The range of muscle fiber composition within any single population, any single ethnic group, any single country, is enormous, often larger than the average difference between two different populations. Two athletes from the exact same country, the exact same city, even the same family, can show dramatically different muscle fiber ratios. Population-level averages exist, but they explain only a small fraction of why any specific individual becomes an elite athlete. This is where East African endurance running deserves a closer, more careful look, because it's one of the most studied and most misunderstood examples in all of sports science. Kenyan and Ethiopian runners have dominated elite long-distance running for decades, and a huge number of the world's top marathoners trace their ancestry to a remarkably small number of specific regions within these countries, not entire nations, not entire continents, but specific highland districts like the Kalenjin-speaking regions of Kenya's Rift Valley. Researchers studying this phenomenon have pointed to a combination of factors: growing up at high altitude, which naturally increases the body's oxygen-carrying capacity over years of development, a cultural tradition of running long distances to and from school from a young age, relatively lean body types common in these specific populations, and, critically, a well-developed athletic infrastructure and coaching pipeline that specifically identifies and develops young distance running talent in these regions. Genetics plays some role in this story, but researchers studying the phenomenon consistently emphasize that no single gene and no broad racial category explains it. It's a specific regional population, shaped by a specific combination of altitude, culture, economic incentive, and training. Systems, not African genetics, as a broad, undifferentiated category, since the vast majority of African countries and ethnic groups show no comparable dominance in distance running at all. One specific biomechanical study helps illustrate exactly how narrow and specific these advantages actually are. Researchers comparing the lower leg structure of Kenyan runners against European runners found that Kenyan runners tend to have measurably lower leg mass concentrated closer to the knee, with thinner calves and lighter feet, a structural difference that reduces the energy cost of swinging the leg forward with each stride, somewhat like the difference between running with a lightweight stick versus a weighted one strapped to your shin. This is a genuinely real, measurable biomechanical advantage for distance running specifically, but it's a trait tied to a specific regional population's typical build, not a broad racial category, and it would offer no comparable advantage in sports built around raw power or explosive short-distance speed, which demand an almost entirely different physical profile. The Kenyan town of Eton, sitting at roughly 8,000 feet above sea level in the Rift Valley, has become such a globally recognized training hub for distance runners that athletes from Europe, North America, and elsewhere now travel there specifically to train at altitude alongside Kenyan runners, hoping to capture some of the same physiological adaptation. That detail alone undercuts a purely genetic explanation. The altitude, the training culture, and the community of accomplished runners to train alongside are all things a visiting athlete from anywhere in the world can access simply by showing up and living there for a few months, which is precisely why so many do. This regional specificity matters enormously, and it's the detail that gets flattened almost every time this topic gets simplified into a race-based headline. Nigerian sprinters, Jamaican sprinters, Kenyan distance runners, and Ethiopian distance runners all have African ancestry, and yet these populations show completely different, sometimes opposite, athletic specializations, explosive power in one case, sustained endurance in another. If broad racial category alone explained athletic dominance, you'd expect uniform patterns across an entire continent's worth of genetically diverse populations. Instead, researchers find highly specific regional and cultural patterns that don't map cleanly onto any broad racial grouping at all. Testosterone is another factor that gets brought up constantly in these conversations, often based on outdated or poorly supported assumptions. Multiple large-scale studies measuring testosterone levels across different population groups have found no consistent, meaningful difference in average testosterone levels between broad racial categories once studies are properly controlled for age, body composition, and measurement methodology. Some earlier studies that reported differences have since faced serious methodological criticism, including small sample sizes and failure to control for confounding variables like body fat percentage and time of day the blood sample was drawn, both of which significantly affect testosterone readings. A frequently cited 1980 study claiming a substantial testosterone difference between black and white American men, for example, was based on a relatively small sample and has never been reliably replicated by later, larger, more carefully controlled research, yet the original claim continued circulating in popular and even some academic discussions for decades afterward, a pattern that shows up repeatedly throughout the history of this topic. A flawed early study generating a claim that outlives the actual scientific support for it many times over. The scientific consensus today does not support the idea of a consistent, biologically meaningful testosterone gap between broad population groups that would explain differences in athletic performance. What actually varies enormously in testosterone, as in nearly every other physiological measure discussed in this video, is the range found between individuals within any single population, a range that dwarfs whatever small average difference might exist between broad population groups. It's worth understanding where the assumption of a clean racial hierarchy in athletic ability actually came from, because much like earlier ideas about race and biology this channel has covered before, it wasn't discovered through careful science. In the 1930s and 1940s, following the visibility of black American athletes like Jesse Owens dominating international competition, a number of physiologists and physical anthropologists rushed to construct theories claiming innate anatomical differences, longer limbs, different tendon insertion points, supposedly denser muscle fibers that would explain black athletic success through fixed racial biology rather than training, opportunity, or individual talent. Most of these specific claims were built on small, poorly controlled samples, and many were later shown to not replicate at all under more rigorous testing. The pattern repeated itself decades later in different sports and different eras, each time treating a specific population's success in one particular athletic discipline as proof of a sweeping innate racial trait, while conveniently ignoring the much larger number of populations and sports where the same supposed pattern didn't hold up at all. It's worth being direct about what actual sports genetics research does and does not support, because this specific topic has a long, uncomfortable history of being misused. There is no credible, peer-reviewed genetic research supporting the idea that any broad racial category is inherently, uniformly stronger, faster, or more athletically capable than another as a biological class. What the actual research supports is something far more specific and, frankly, more interesting. Certain, narrow, well-defined populations, shaped by particular combinations of ancestry, environment, culture, and training systems, show statistical tendencies towards specific athletic specializations. Jamaica's sprinting dominance and Kenya's distance running dominance are two of the most studied examples of this in the world, and in both cases, researchers who've spent their careers studying the phenomenon point to training systems, cultural investment, and specific environmental factors as doing most of the explanatory work, with genetics contributing a real but modest and highly specific piece of the picture, not a sweeping racial explanation. Strength sports specifically, rather than sprinting or endurance running, offer maybe the clearest evidence against any single racial pattern in athletic dominance, because the countries that actually produce the world's top raw strength and power athletes are remarkably diverse and don't fit any simple pattern at all. Olympic weightlifting world records and championship medals over the past several decades have been dominated overwhelmingly by lifters from China, Iran, various Eastern European nations, including Bulgaria and Georgia, and Central Asian countries like Kazakhstan and Uzbekistan. Competitive powerlifting, a related but separate strength sport measured in raw squat, bench press, and deadlift totals, has produced elite champions from an even broader mix of countries, including the United States, Russia, Ukraine, and Lithuania. If there were a clean, sweeping racial hierarchy for pure strength, you would expect this pattern to look nothing like it actually does. Instead, weightlifting dominance tracks almost exactly the same way sprinting and distance running. Dominance does toward specific nations and regions with deep, dedicated, government-supported training systems for that particular sport, regardless of which broad racial category those nations happen to fall into. There's a term researchers use for part of what's actually happening here: selection and funneling. Countries and communities that build strong athletic infrastructure around a specific sport, dedicated academies, coaching pipelines, cultural prestige attached to succeeding in that sport, end up funneling enormous numbers of talented young people into that one specific pursuit, generation after generation. Jamaica, a country of under 3 million people, has built an extraordinarily deep sprinting development system. Youth athletics is treated as a major cultural event, complete with nationally televised high school championships, known locally as champs, that draw crowds rivaling professional sporting events elsewhere, packing an 18,000-seat national stadium for a schoolboy and schoolgirl track meet, with top performers scouted directly into elite training programs and university athletic scholarships from their early teenage years onward. That kind of concentrated, sustained cultural and institutional investment produces elite performers at a rate that a country with an equally talented gene pool, but without that same infrastructure, simply never will. The genetic starting material might be broadly similar to what exists in dozens of other countries and populations worldwide. The system built around identifying, training, and elevating that talent is not, and that system explains an enormous share of the outcome. Childhood development conditions add yet another layer that rarely makes it into the simplified version of this story. Nutrition, healthcare access, and general living conditions during childhood and adolescence measurably affect adult height, bone density, and overall physical development, all factors that feed directly into athletic potential, regardless of underlying genetics. Populations that have experienced historical patterns of poverty, malnutrition, or limited healthcare access show measurable effects on average adult physical development, effects that can shrink or widen depending entirely on changing economic and social conditions over time, not on any fixed genetic ceiling. Researchers studying height trends, for instance, have documented substantial increases in average adult height across many populations worldwide over just the past century, changes far too fast to be explained by genetic evolution and instead driven almost entirely by improved childhood nutrition and health care. That same principle applies to athletic development broadly. Populations' average athletic outcomes today reflect current training systems, current economic opportunity, and current childhood development conditions layered on top of a genetic baseline that, as this video has shown again and again, varies far more within populations than between them. None of this means individual genetic variation doesn't matter. It clearly does. Elite athletes across every sport and every population studied consistently sit at the extreme statistical tail of numerous physical traits: height, limb proportions, muscle fiber composition, cardiovascular efficiency, and yes, specific gene variants that offer small individual advantages. But that variation exists richly within every population on Earth, not cleanly between broad racial categories. An elite sprinter and a middle-of-the-pack recreational runner from the exact same ethnic background can differ more genetically and physiologically than the average difference between two entire populations. Elite athletic performance is what happens when a rare combination of individual genetic traits meets exceptional training, opportunity, nutrition, and often no small amount of pure chance. Not when one broad population wins some sweeping biological lottery that another population loses. So when you go back to that original comparison, DNA from elite athletes across the world's population groups laid out side by side, hunting for the genetic explanation behind a supposedly massive uniform racial gap in strength. The actual data doesn't support that gap existing in the way the headline implies. What it supports instead is a patchwork of narrow, specific, well-documented stories. A highly localized genetic and cultural phenomenon in Jamaica, a different one rooted in altitude and running culture in Kenya's Rift Valley, individual-level genetic variation that swamps population averages almost everywhere. It's been carefully measured, and a heavily publicized gene that turns out to explain only a small sliver of the picture once researchers actually test it directly against elite performance. That's the real gap researchers keep finding when they run these numbers properly. Not a gap between races, but a gap between the popular myth and the actual, carefully measured science. Genetics matters, but it operates at the level of highly specific individuals and highly specific narrow populations, shaped by particular histories, not at the level of broad, sweeping racial categories invented for entirely different purposes centuries before anyone had the tools to test any of this directly. Go back to that stopwatch in Kingston and that teenager running to school in the Rift Valley before sunrise. Neither one of them is the product of some sweeping racial destiny written into their DNA at birth. Each one is the product of a remarkably specific set of circumstances, a training system built over decades, a culture that celebrates and invests in exactly this pursuit, an altitude or an infrastructure or a coaching lineage that happens to exist in that particular place and nowhere else quite like it. Layered on top of individual genetic variation, that exists just as richly in populations that have never produced a single world-class sprinter or marathoner at all. The stopwatch doesn't measure race. It measures the outcome of 1,000 specific traceable factors, most of which have nothing to do with the broad category so many people still reach for first. There's a broader lesson sitting underneath all of this, one that applies well beyond sports. Whenever a complex outcome, shaped by dozens of interacting factors, gets flattened into a single, simple explanation tied to a broad category like race, the actual science almost always turns out to be more specific, more localized, and more interesting than the simplified version. Athletic performance is one of the clearest examples of this pattern precisely because it's so visible, so measurable, and so heavily documented across decades of serious research. And every time scientists have actually dug into the details behind a supposed racial gap in strength or speed, they found the same thing. A tangle of highly specific regional, cultural, and individual genetic factors, never a clean, sweeping biological hierarchy. If this changed how you think about where athletic dominance actually comes from, like this video, and subscribe, because we dig into the real science behind these headlines every single week, not the version that spreads easiest online. Drop a comment with which sport or population story you want us to dig into next. And if you know someone who still believes one race is simply built stronger than another, send this their way, because the actual research paints a far more specific, far more interesting picture than that old, tired assumption ever did.